Hello. Today we will be discussing regarding this non-Mendelian inheritance. A Mendel considered 1:1 relationship between character and factor or gene. That is, a single character is controlled by a single gene or factor, but later on it was observed that this 1:1 relationship is not always necessary, and there may be cases where more than one factor or genes control a single character. Further, one factor or one gene may be involved in more than one character, that is, pleiotropic gene. This hypothesis that a single character may be controlled by more than one factor and a single factor may affect more than one character was first put forward by BT1 and is called BT1 factor hypothesis or BT1 factor interaction. The interaction modify normal F2 ratio, that is 3:1 in monohybrid ratio and 9:3:3:1 in dihybrid crosses. So, whenever there is pleiotropy or when there is a single character governed by more than one gene, always the inheritance doesn't follow the Mendelian pattern. So, we call this kind of inheritance as non-Mendelian inheritance. So, one of the non-Mendelian inheritance is incomplete dominance. In Mendelism, we have seen that one allele is dominant over the other, but here none of the allele is dominant over other 100%, and the F1 hybrid have an appearance somewhat in between the phenotypes of the two parental varieties. And the example is the snapdragon flowers. Normally, there are two color of the snapdragon flower, that is red and white. So, in case of red, the genotype is homozygous, that is capital R and capital R, and in case of this white phenotype or white flower plant, we have small r homozygous, so called recessive, and whenever a cross is made between a red colored snapdragon with white colored snapdragon, we find that in F1 all the plants are neither white nor red, rather one intermediate color, that is pink. So, here this is the white parent and this is the red one, the flower color is red, and when in F1 generation we find all the plants are of pink, and when it is crossed with another white type, what we get? We get 1/4 as white half as pink, 1/4 as red when a selfing is done between two plants of F1, but when we go for a test cross, we find half are of pink type and half are of white type. Similarly, there is another example where both the alleles are expressed in heterozygotic condition, and in heterozygous individual both the alleles shows their presence. So one of the example is the blood type. This type of inheritance is called codominance, where none of the allele is dominant or recessive, rather they show their presence in heterozygotic condition. In case of blood group, the type 1, where you will have IA, capital A or capital A. Similarly, in B also you will find this is like homozygous, or maybe only in one this B is present, B allele is present, and in type AB both the alleles A and B are present, and in another case like in O group none of the alleles are present. Let us see one example. Suppose one homozygous male of type B or blood group is crossed with a heterozygous female of type A, then we find different combinations. Like in case of female we have two gametes, one is with antigen A and another with non. Similarly, in this case, in case of male, both the gametes have the same kind of allele, and as a result we expect 50% are of AB type and 50% are of B type. And another problem, let's see, one male without these alleles is crossed with female type with AB. Then for the female in the gamete we have both the alleles, so 50% are of A type and 50% are B type, whereas both these are of heterozygotic in nature. So if a boy has a blood type O and his sister has blood type AB, what are the genotypes and phenotypes of their parents? See, the boy has type O, that means it does not have any kind of these antigens, and the girl has type AB, so both the alleles are present. Now, if the parents have like heterozygotic A and B types, then you expect this kind of phenotypes. Now there are certain other traits which doesn't follow the Mendelian inheritance pattern. These traits are linked to sex chromosomes. So these traits we call them sex linked traits, and these sex linked traits are located on sex chromosome. In case of human being, these traits are located on either X or in Y chromosome. In case of human being, for females, the genotype is always XX, whereas in case of male, the genotype is always XY. So many sex-linked traits carried on X chromosome. Now let us see the eye color in fruit fly. Sex chromosome. So in fruit fly, the female has two X chromosomes, whereas in case of male, there is only one X and another Y chromosome, and this fruit fly eye color is associated with the X chromosome. So suppose a red eye male is crossed with white eyed female. So capital R is for dominant red color, red allele, and small r is for white. Then this Y chromosome doesn't carry any trait. So RR is for red, RR is also for red, whereas homozygous small r is for white eye, and XY is male and XX is female. Now %, will be red eye female, whereas 50% will be white eyed male. In sex linked traits, there are certain gene diseases associated with the X chromosome, like that of hemophilia, where women are carriers and men have the phenotype more often because this is associated with the X chromosome, and in case of the male there is only one X chromosome and it's a recessive in nature. So whenever this gene is present in the X chromosome, the male used to suffer from the disease, whereas in case of female, where there are two X chromosomes and in one if the allele is recessive and in other one it is dominant, then the female is a carrier. It can inherit the disease from one generation, or it can propagate the disease from one generation to other. However, it doesn't show any kind of symptoms in the female. Now let us see some breeding problems. Suppose a tall and a dwarf pea plant is crossed. What we get? We get always a tall plant that is heterozygous in nature in F1 generation. However, in F2 generation, we get a 3:1 ratio, which is in case of Mendelian inheritance. However, in genotypic ratio, it is always 1:2:1. One is dominant homozygous, two are heterozygous, and one is homozygous recessive. So these are some of the examples we have seen in case of non-Mendelian inheritance. However, there are certain other phenomenon associated with these non-Mendelian inheritance, like these lethal factors, these collaborator genes, then these phenomenon of epistasis, uh, which we will be discussing in the coming class. Thank you.